So now again I am going to explain you another spotting, that is the study of pollen germination and growth of pollen tube in a pollinated pistil in Portulaca. Okay. So the experiment is to study the pollen germination and the growth of pollen tube. In an angiospermic plant, the flower is the reproductive organ, as you know. It contains the male and the male reproductive organ is known as stamen and the female reproductive organ is known as carpel. The stamen contains the three parts, that is your anther, connective and filament. Anther contain the male gametophyte, that is your pollen grain. Similarly, the carpel, it contains the three parts, that is the receptive part that is your stigma, style and ovary. Ovary contains the embryo sac. So pollen grain, which is the male gametophyte, when falls on the stigma, it's germinate. So this is the structure of a carpel. This is your ovary, this is your style and this is your stigma, and pollen grain, which is the male gametophyte. By the process of pollination, when it is received by the receptive part of the carpel, that is your stigma, it's germinate. The pollen grain has the part, that is the two layers or theca, that is your exine and intine, and it contains the vegetative cell and generative cell. The pollen grain, when falls on the stigma, give rise to pollen tube from the vegetative cell. Along with the cell of the stigma and the style, it enters and moves towards the embryo sac by dissolving the cells or the tissues which are present at the style at the stigmatic region. So this experiment is based on it, that when pollen grains falls on the stigma, what are the changes which is been observed on. The pollen grains contain the pollen tube contain the vegetative cell and two male gamete when it is completely been matured enough. So the pollen tube, when it germinates and moves towards the embryo sac, the generative cell divides mitotically and give rise to two male gamete. So this pollen tube, when germinate, it contains, when it reaches at the embryo sac, it contains this vegetative cell and the two male gamete. This is the conclusion which you people will write it down at the end. So whole experiment, the slide will be there on the table. The specimen will be given to you. Mom is going to explain you now. and you will have to write it down, the identifying character with the diagram explaining at the end the conclusion. The germinating pollen tube, the germinating pollen tube contain vegetative cell and the two male gamete.
Hello students, today I am going to demonstrate how naturally pollen grain germinated on stigma surface. And you know that stigma is the part of pistil or carpel. So look in China rose flower, here these are anthers, yellow color anthers, and here at the top there are deep red color stigma surface. So this is a part of carpel. So you have to take any small size flower like Portulaca. And this is Portulaca flower. So you have to open this flower using a forceps, and here you will get male and female part. Only you have to take the stigma and style part. Using forceps, look here, this part is stigma and this is style. And you have to remove this part and keep on a slide. Okay. Now you put a drop of water. Next, you have to put a staining material, that is safranin. You know, color of safranin is pink. So put a drop of safranin here. Okay, and keep for a minute. Then use blotting paper to soak excess safranin. Look here, you have to remove safranin using a blotting paper. So on both sides you have to remove, because your slide should be neat and clean. Both safa rakhiyak padibo. Next, you have to put a drop of water to wash away excess amount of stain. So you have to use brush, not needle. So remove excess amount, all water you have to remove. Next, you have to put a drop of glycerin. Why you need glycerin? Because glycerin has same isotonic solution, so the specimen will remain fresh. Nale dry hejibo, tenu gote drop glycerin eti pakebo. Glycerin pakela pare cover slip kadibo, and carefully you have to drop cover slip. I'll show you how to carefully have to drop. Just keep this for cover slip 60-degree angle with the support of a dissecting needle. Okay, and gently you have to release this needle. This process is called mounting. So after mounting, you have to place it under microscope at low power. So when placed in under low power, you have to see through the eyepiece and adjust this course. Okay, and observe the specimen. So it should be focused on the sufficient sunlight. Otherwise, it will not visible clearly. So you have to focus on sunlight. So that's all about your pollen germination technique. How to carry out this experiment to show how stigma on the stigma surface pollen grain germinates. Thank you.